Okay, in this first video, I'm going to be talking about the light cycle of photosynthesis. And as I'm sure you know, the light cycle takes place inside of the chloroplast, which is one of the organelles commonly found in plants, algae, etc. So, before we can really start talking about what's happening, we need a basic idea of the structure of the chloroplast molecule. So basically you've got two membranes here, an outer membrane, an inner membrane, some space between them. Inside this inner membrane, you've got this series of stacked discs. Each disc is called a thylakoid, and the entire stack of them is called a granum, or grana for plural. Now, looking at each individual thylakoid, you have the thylakoid membrane, and that's basically what this is another phospholipid membrane. And then the space inside of it, which is called the thylakoid space, or sometimes called the lumen. All around all of the grana, we have liquid, and that liquid is called the stroma. So now that we know the basics of it, we're going to zoom in on just one of these thylakoids, and that's pretty much what I've drawn on this screen for you. So let's go ahead and start talking about that. I'll move this so you can see it better. And so what we have here is one single thylakoid, and embedded in the thylakoid are these protein reaction centers. And these reaction centers are composed of a lot of different pigment molecules. And within it, this first one that we are going to start looking at is what we call P680. And it's called the P680 molecule because it really absorbs the wavelength of light at 680 nanometers very well. Within it, you've got a bunch of pigment molecules, and then you've got these two special chlorophyll A molecules, and they're the ones that are ultimately going to be giving up their electron to this thing, which is our primary electron acceptor. Now, in order for all of this to start, you do, of course, need the sun. So, the sun shines its light down on the light harvesting complex here, this area of all the pigment molecules. And what happens is, as the light bounces around inside there, all of that energy is eventually collected and passed to those two special chlorophyll A molecules. And as they get full of this energy, they start getting so excited that they actually give up one of their electrons, which is what we call being oxidized. And so to be oxidized is to give up an electron. So it passes its electron to the primary electron acceptor, which in turn is going to pass it to a chain of other proteins that I have not drawn yet. But we're going to hold the thought on this electron. Before we get there, we need to worry about these chlorophyll molecules because they are no longer stable. If you give up an electron, you're going to need to replace it from somewhere else. And so that's where all of this water that's in the plant comes in handy. The water molecule is, of course, made of hydrogen and oxygen. The hydrogen is going to sacrifice its electrons, so that way the missing electrons from the chlorophyll molecule are now replaced. These leftover hydrogen molecules are then free to float all around the stroma. So I'm going to draw a few of them out here in the stroma. And then plenty of them are here in the lumen, our thylakoid space. So I'm going to draw a bunch of them here. Now, how they got there, we'll get to that. But right now, it's enough to know that there are a whole bunch of these hydrogen ions, also called protons. You could call them either way. Hydrogen ions or protons, they are the same thing. The inside of this area is totally full of protons. Now, that takes care of the hydrogen, but this oxygen is left over. Oxygen will not travel alone by itself. It's not stable. It needs to be bonded to something else. But if this happens to more than one hydrogen, two of them specifically, those two spare oxygens can partner up, and they are going to be released into the atmosphere as waste for the plant. Of course, that's not waste for us. That's where we get the oxygen that we are breathing. So every time you are breathing, you are actually getting the oxygen that was split out of these water molecules when the hydrogen donated its electrons over here to these chlorophyll molecules. So everything that we've looked at so far, this business with the excited electrons at the P680 center, and these water molecules helping replace that loss, is all called Photosystem II. Now, you're probably wondering why it's called Photosystem II and not 1, and the reason why is because this was was actually discovered second. Photosystem I exists, but it happens later. It actually can't even happen without Photosystem II having happened first. But we discovered the other one first, so we called it the photosystem. And then once we found this one, then, well, that's Photosystem II. So that's the basics of how it got its name. Let's go back to our excited electron. Now, meanwhile, as I said, there are plenty of other proteins floating around in the thylakoid membrane. So I'm going to draw a couple more of those in. The first of these that we're going to look at is called plastoquinone, and I'm going to go and abbreviate it as PQ. And what ends up happening is this electron that is full of all of this high energy is going to get passed to the plastoquinone. Now, the plastoquinone at this point has gained an electron, so we say it's oxidized. The primary electron acceptor, it gave up the electron, we say it's reduced. So this is a redox reaction because one thing is getting reduced, the other thing is getting oxidized. You can always remember which is which because if you remember the term oil rig, that abbreviation, oxidation is loss, reduction is gain. It tells you what happens to the electron. So if you remember oil rig, you can remember which molecule is which in a redox reaction. Now, back to the plastoquinone. This molecule is a protein that can change shape, and it is going to change shape. It's going to use some of that energy from that electron, and it is going to do some active transport. Right now, we have a higher concentration of protons inside our thylakoid and a lower concentration outside in the stroma. So some of this energy from this electron is going to be used to pump the hydrogen from the stroma into the lumen. Now, that did not use up all of the energy from this electron. This electron has more energy to give. So from there, it's going to pass it on to another protein called the cytochrome complex. And again, this one is losing an electron, so it's oxidized. The cytochrome is gaining an electron, and so it is reduced. And then cytochrome is going to do the same thing. It's going to pass it on to yet another protein, plastocyanin, which I'll abbreviate PC. And the plastocyanin is going to go and take the energy and do exactly what this first one did. It is going to do some active transport, and it is going to pump some of these hydrogen ions into the thylakoid space. Now that still has not used up all of the energy from the electron. So this electron is going to be used to do something else. But once again, we're going to hold that thought. Now, all of this that I've listed so far with this series of proteins that have been doing this active transport are what we call the electron transport chain. Sometimes you see this abbreviated as the ETC. This is our electron transport chain. And as I'm sure you've noticed at this point, the inside of the thylakoid is really getting full of hydrogen. So you can't keep pumping it in and not expect it to go anywhere. Besides, the going somewhere of the hydrogen is the whole point of this entire reaction. So there is yet another protein embedded in the membrane. I'm drawing it down here on the bottom, but there's no reason why it has to be on the bottom. All of these proteins are just kind of floating freely and randomly kind of bouncing around in that phospholipid bilayer that makes up this membrane. But this one that I've drawn down here is what we call ATP synthase. And it is a very important molecule. It's kind of like a revolving door. So what ends up happening is these hydrogen ions are going to go and diffuse out of the thylakoid space. So this is a matter of facilitated diffusion. They are going through the ATP synthase. But the ATP synthase works kind of like a revolving door. And so as these pass through, we are calling this process chemiosmosis. I will write that here for you. Chemiosmosis. And as the hydrogen flows through, that is causing my ATP synthase to revolve like a revolving door. And so the force that is making this thing actually revolve is what we call our proton motive force. It's actually causing this revolution. And now, as you know, things that turn, those are engines. You can go and take that energy and use it to do some other kind of work. So in this case, what we are going to do with that energy is this protein is going to emit one adenosine diphosphate, ADP, and also another phosphate molecule, and those will go into the ATP synthase. What is going to pop out the other side is a high energy molecule of ATP. So basically this is stored energy that's going to be used later on when we get to the Calvin cycle. So ATP synthase revolves because of the proton motive force, and that couples the ADP and the phosphate and forms the molecule ATP, adenosine triphosphate. So basically this is now energy that is stored. So all the energy that originally came from up here at Photosystem II, it was captured by the chlorophyll molecules, used by the electron transport chain to do some active transport, is now being used again to produce this ATP. So basically we are transforming light energy here to chemical energy there. So this is a very large portion of where this energy that's going to be eventually turned into sugar comes from. But we are not yet done, because remember, we forgot this electron up here. So let's go back to that electron. Meanwhile, up here, there is not just one, but two of these light reaction complexes. So I'm going to draw another one of them here in place. Once again, it's full of pigment molecules, and it's got the special chlorophyll molecules, and it's got the primary electron acceptor. And just like this first reaction center that we drew, the sunlight is shining upon it. So this zigzaggy line is representing my light energy that is now striking this particular reaction complex.